Stu? What's up, man?
What up, Ben?
Hey Stu, what's up?
Hi, Stu. You feeling better? Good. Why don't you come in and we'll make up the lab. What was that, Stu? Did you miss anything important during the lab? Okay, guys, let's review what you should know before starting this lab.
All living things are made of cells, including trees, cats, mushrooms, and single-celled organisms. Some cells are eukaryotes, which are made of organelles. On the left is a diagram of a plant cell, and on the right we have an animal cell.
We're going over the materials list before we get started. Let's begin. Gelatin. Powdered, this is our source of protein. We've diluted in warm water and a reagent, Bioret, to test for its presence. Reagents, we need water. Water is to dilute our other samples in and to serve as our negative control. Corn syrup, any light or pancake would do. We've diluted a little in water and Benedict's is the reagent. This is why we will need the hot plate. Assume the hot plate is hot until you know that it is not. Cornstarch, another carbohydrate, a complex one this time. Just put the powder in a little bit of water and iodine to test for its presence or not. Vegetable oil is our lipid, our fat, and we will be using paper bags to test for its presence or absence, since we don't have Stu Dent 2 or 3 or 4 today. The first goal of today's lab is to create a set of standards for each test. Here, the protein test, negative control, positive control, Bioret protein turns lavender. Here, the negative control and positive control for the simple sugar, that was the corn syrup test. This is the negative, this is the positive. The positive and negative control for the starch test, since clear and brown is what the reagent normally looks like, and in the presence of starch it turns black. Then for the fat test, you can tell which one looks like a greasy French fry bag, can't you? Then once we have used these to create this, then you will be given the unknown solution, which contains 2 of the 4 test subjects. You must tell me which 2 are in here. Okay, student scientists, it's now lab time. No, not too much. Let's get started. So what we're going to do for you today, Stu, is we're going to test this unknown. It contains 1 or more of the biomolecules that we have been working on. What we need for that is some test tubes, the paper for the fat test, and our reagents, the Bioret, the Benedict's, and the iodine. But before then, and the boiling water. Before then, what we need is to put on the personal protective equipment. Great, let's put on the gloves because a few of these reagents do contain chemicals which could be irritants. Not a big deal, but better safe than sorry. Great, let's get started. The first thing we're going to do is this. You are going to put 2 dropperfuls of the unknown in each of the test tubes. So give it a shake. You're going to pull up a full dropper, put it in, and then do it again. That's roughly going to give you about 5 mils per tube. So there you go. single drop right here. Just one drop. Cool. Shake it around, smear it a bit, shake off the rest. Now, does that look greasy like this does? Alright, we have just confirmed that this unknown has no lipid. Your first test is done. Good job. Second test, protein. Now, this is using the Biuret reagent. So in this first tube right here, give me 10 drops of Biuret right here. All right. Okay. I have a clear appearance here, and now it's kind of light. It's kind of lavender. It almost looks like this. Not quite. Give me a few more drops. Okay, now I think we can definitely see a difference, and that comparing between the negative and the positive control, we can say this is definitely positive for protein. Wouldn't you agree, Stu? Great. Next test is the simple sugar test, and that is 10 drops of this into this tube. Great. Now, we need to heat this. So, since this water bath is boiling, I'm going to fly it over here. The heat's required for the reagent to react with the sugars and make the characteristic Sunny D tang reddish brown to orange precipitate. We might need to come back to this. Here we're going to do starch test. If you will put, let's just go 5 drops into here. Great. Oops. Okay. Brown is a negative reaction. Black, jet black, and thick is a positive reaction. So, offhand, I would say, with no black precipitate at all, that this is a negative starch reaction. Wouldn't you agree, Stu? I think we're beginning to see a color change. We're definitely not light blue or Easter egg blue anymore. As we set up the unknowns, different types of unknowns and different amounts, let's say I used powdered milk, or I used powdered gelatin, or I use corn syrup, the amount that I put in will make a difference in how quickly the reaction happens and what it looks like. A little more or a little less drops in the test would make it a bigger deal. And I think we're almost there. It's not quite as robust and in your face as that, but it's definitely not like the negative. So, let's recap. Did it contain lipids or fats? No. Did it contain starches? No. So it did definitely contain protein, and here we go. Simple sugars. And that would be the identity of the unknown, which in this case, I will admit, is dry powdered milk. Good job, Stu. So, now that you've seen the standards and you've done the unknown, you should now be ready to completely fill out and turn in your lab packet. Okay, so now what we need to do is clean up, take off our personal protective equipment, and we should be prepared and ready to go. Hey, Stu. Before you go, why don't we see if you are prepared to deal with these on, I don't know, some standardized test. Having the knowledge and doing the lab is one thing, but being able to work it out on a test is really very different. So let's see if you're ready.
A student drops a few drops of iodine on an unknown piece of food. The sample turns dark purple-black. Which molecule is present? A. Lipid, B. Starch, C. Simple sugar, D. Protein. Use the table above to answer this question. Dr. Strangelove conducted an investigation using bioreagent to determine the presence of proteins in different foods. The results are shown in the data table above. According to the table, which foods tested by the students contained proteins? A. Honey and potato. B. Potato and chicken broth. C. Cottage cheese and water. D. Cottage cheese and yogurt. A scientist performed 3 tests to identify several unknown substances. The results of these tests are summarized in the chart below. According to this chart, what kind of molecule is in substance 2? A. A lipid. B. An amino acid. C. A nucleic acid. Or D. A carbohydrate.